So the session focused on alpha-synuclein as a target is focused on this idea that because alpha-synuclein is genetically associated with Parkinson's disease, and because it accumulates in Lewy bodies in the brains of patients with Parkinson's, and of course patients with other disorders as well, this all suggested that maybe we need to interfere with the aggregation of this protein. There's multiple strategies being tried. One is the immunotherapy, so antibodies that will recognize aggregated forms of synuclein or soluble forms, but will interfere with the aggregation. So this is one strategy. Another strategy is using small molecules that are capable of interfering with the aggregation process. And there's also another strategy that seems to make a lot of sense also based on what we know from genetics, and that's to try to reduce the production of alpha-synuclein. We think that the problems arise when there's too much accumulation of alpha-synuclein. So if we are able to reduce the production of the protein, we may be able to avoid this threshold after which the protein will start to misbehave, to aggregate, and to become problematic. But again, it's important to keep in mind that this is testing only one side of the problem of this hypothesis, and that we need to not forget that maybe it's also a loss of function. So we need to also consider strategies in the future and to test this idea that maybe we need to increase synuclein function. The problem there is that we still don't understand synuclein function very well, and so it's difficult to increase the function of something we don't know exactly how that is working. But in the future, hopefully we'll be also learning more about that. So the current efforts aimed at lowering the spreading and aggregation of synuclein are focused primarily on two approaches. One is the immunotherapy, as I mentioned, so trying to use antibodies to interfere with the aggregation, and because we think that the spreading of these toxic forms and aggregated forms of synuclein involves proteins going from one cell to the next, antibodies may be able to capture and catch these forms of the protein while they are in between cells, thereby interfering with the spreading. The other strategy is reducing the production using antisense oligonucleotides. This is being tested by different groups and with promising results, but we'll have to see when we have clinical trials to test these molecules, how they behave.